If you're a Year 13 A-level biology student who's been studying aerobic respiration, specifically oxidative phosphorylation, then this video is for you. So the first thing we need to look at is the fact that the inner and the outer membrane are both made up of phospholipids. Now this is a very basic diagram here to show you a membrane with all the important proteins that are involved. Now, the part closest to me here is actually showing you the mitochondrial matrix. So remember, in that mitochondrial matrix, we have had the link reaction and we've had the Krebs cycle. Well, what's really important is for us to take a quick recap over each of the different processes that have led to the end part, which is the oxidative phosphorylation. So aerobic respiration involves glycolysis, the link reaction, and the Krebs cycle. And the most important thing is about the products, the things that come out of each of the different stages. So when we look at the output for each of the different stages before oxidative phosphorylation, we can see that glycolysis gave us 2 reduced NADs. The link reaction gave us 2 reduced NADs. The Krebs cycle gave us 6 reduced NADs and 2 reduced FADs. Now that is a whole load of coenzymes that are required to take us to this last part in aerobic respiration. So what you can see from my diagram here is that I have very different proteins which are involved in this oxidative phosphorylation in the inner membrane. Now, the one on the left-hand side is called ATP synthase. So ATP synthase, ASE is an enzyme, synth to make the synthesis, is going to synthesize ATP. But to be able to synthesize ATP, first, we need to go over to the proteins on the right-hand side. So you can see there's quite a few proteins. We call them the electron transport chain. And what's really important for us to understand is that our oxidation reaction that was happening before to the molecules was resulting in us having reduced NAD and reduced FAD. Now, those coenzymes, reduced NAD and reduced FAD, metaxes, are going to deliver hydrogen ions and electrons to the electron transport chain. Now, electrons have got energy to them. So what happens is as the electrons are transferred through the electron transport chain, what happens is the energy that they have is actually being used to pump hydrogen ions against their concentration gradient, active transport, from a low concentration to a high concentration into the intermembrane space. So we're going across the inner membrane, so from the mitochondrial matrix into the inner membrane space, which is between the outer membrane and the inner membrane, and therefore creating a concentration gradient. Now, it's important to understand that with the electron transport chain, every time the electron is moving through the chain, it is losing energy as the hydrogen ions are being pumped. Now, the reduced NAD will come to this first protein. The reduced FAD comes to the second protein. Each time it is going to result in hydrogen ions being pumped. So the hydrogen atoms can be separated out into, so they can be lysed into hydrogen ions and electrons. And it's those electrons that are free to move through the electron transport chain. Now, the only way they can move through is if the next proton, the only way they can move through is if the next protein is freed from having the electrons. So what's important for us to understand is in our electron transport chain, it is not only proteins that are involved. Oxygen is known as the last electron acceptor. So if there is no oxygen present, there is no last electron acceptor. Therefore, the electron transport chain cannot function. When the electron transfers from the last protein to the oxygen, it can bind with the oxygen and two hydrogen ions and therefore result in the production of water. So water is a product that is produced by aerobic respiration because the oxygen is the last electron acceptor, and that combines those electrons, the oxygen and hydrogen ions combine together to produce the water at the end. So what's the point in pumping these hydrogen ions across into the intermembrane space, into the space between the cristae? It is going to create a high concentration, and that high concentration is going to result in a gradient across the membrane. So the hydrogen ions are going to diffuse down through the ATP synthase, down their concentration gradient. Now, as they do that, as the protons make their way through the ATP synthase to generate the energy that is required to join the adenosine diphosphate, ADP, with the inorganic phosphate, therefore creating ATP. So the production of ATP is driven by the movement of those hydrogen ions through that ATP synthase. Now that process, the ETC, the electron transport chain, and the movement of the hydrogen ions to produce the ATP is known as chemiosmosis, generating a concentration gradient so that the hydrogen ions can then diffuse through the stocked particle ATP synthase is chemiosmosis. Osmosis, normally we talk about water, but we're talking about movement from a high concentration to a low concentration through a partially permeable membrane. Well, chemiosmosis is the chemical, so the proton hydrogen ions, moving down its concentration gradient through the stocked particle, which is known as ATP synthase. And that is all about the production of ATP. So it's important to understand that the reason that reduced NAD and reduced FAD are produced is for that production of ATP. Now, for every reduced NAD, that equates to 2.5 ATP molecules. And for every reduced FAD, that equates to 1.5 ATP molecules. So therefore, for every glucose molecule, it results in 32 ATP molecules being produced. Now, depending on where you do your research, you may see where they say that it's for every reduced NAD is 3 and for every FAD is 2. And that number can be anything between 32 to 38. But it's important that you look at your own specification and what it is that you need to know. We did believe it was 3 and 2. However, as we advance our knowledge, as we continue to learn more and more, we therefore can amend our theories, and now it's believed it's 2.5 and 1.5. But the key thing to remember is that some ATP molecules need to also be used. They need to be used in glycolysis, they need to be used in active transport, they need to be used in other ways, and therefore the total number of ATP molecules that you get at the end is never quite the same number. It is all dependent on the situation.